Hello, everybody. So my talk will span from basic research to a potential clinical application, and therefore it is organized in two parts. I'm first going to show you how we built a model for the information processing of the Epo receptor, and then how we link this to cancer. And in cancer, anemia is a problem, and therefore we then combine this model here on the cellular scale with the body scale model in order to, in a patient individual way, potentially improve the treatment of anemia. So we're talking about red blood cells. This is what lacks in anemia. And the production of red blood cells in itself is really an interesting system. So you start with some kind of stem cells, and then there are a number of lineage decisions. And if you go this direction here, you end up as a red blood cell. And there is one stage here in between where this further decisions strictly depend on one hormone, erythropoietin, short Epo. If this does not see Epo, the cells just die. If it sees Epos, it survives, proliferates, and then finally differentiates. This system in itself is a nice control system because there is a sensor in the kidney that senses whether there is enough oxygen in the tissue or too much. And depending on this, Epo is produced here in the kidney. And so if you have too much, then it goes down. If you have too small values, then it goes up. These red blood cells live around 120 days. That means they have a fast turnover compared to other cells. And that means that we have a basic level in some funny units of Epo in the plasma. And if you need a lot of blood, then this can go up by 3 orders of magnitude. And what we wanted to understand in the first part now is how the receptor system can deal with these huge differences in extracellular concentrations. And there are in principle 2 possible scenarios. So if you map the ligand concentration to the receptor activation, it could go somehow in saturation, but of course it would be more efficient if you have a kind of linear relationship between this normal condition and the condition where you need a lot of oxygen. So, and what we now want to do is we want to build a dynamic model for the Epo receptor and processing of this hormone and understand based on this model how this broad ligand range can be dealt with. From a biological point of view, there are 4 different possible solutions to the problem. And here on the left-hand side, it's always shown the case where there is few Epo, the normal level of Epo, and here is a high level of Epo. So the first possibility, abundance, would be just to have a lot of Epo receptors. The second one, we call it mobilization, is the following procedure. Is it known that 90% of the Epo receptors are not at the cell membrane, but in intracellular vasculars? And mobilization would mean if a lot of Epo comes, then these receptors are mobilized and brought to the cell membrane. The third possibility is recycling. It is known that these Epo receptors undergo endocytosis and can be recycled. And if you do, and thereby Epo can be degraded. And then that would mean if you do this in a fast manner, you could also see huge differences of this concentration. And the final one, turnover, is similar to mobilization. The difference is that we don't mobilize it, but that we have a constitutive turnover of the Epo receptor to sample the extracellular concentration of Epo. So this first possibility, that can be ruled out experimentally. There are techniques to count the number of receptors on the cell membrane, and the result is it's in the order of 500. And this, of course, is not sufficient to see really differences of 3 orders of magnitude. The 3 others, so therefore this one is out, the 3 other possibilities are not so easily to disentangle experimentally, and therefore we built a model to describe this process and figure out which of these mechanisms are at work and how the translation of the information from outside the cell to receptor activation takes place. Yeah, building such a model always relies a little bit on biological intuition, but it's also a little bit trial and error tinkering around. And what we came up with is that we need these 2 mechanisms to describe the data I show you in a second. And so this is how the model now looks like in a little bit more detail. So here we have the constitutive turnover, and here we have this recycling. Epo can bind with the Epo receptor to form this complex, and they can unbind again, and then Epo can be degraded inside the cell or to the outside the cell, or Epo can go back in this recycling process to the extracellular space. Yeah, what was measured experimentally are the extracellular components of Epo. Epo bound to the receptor at the cell membrane and the compartment inside the cell. I will show you the equations in a second. So these are the data points. Error bars are pretty small since this is a very, very isotopic labeling, very dirty measurement, but it produces really nice data. And with this model, then we were able to describe these data. There were, of course, a lot of data sets more, but this is just as an example. Yeah, I will not go into detail about this slide. So we will spend a lot of work on the question whether parameters are identifiable from data or from models fitted to the data, because only then predictions are reliable. And we use a method that's called profile likelihood in this context. And the message here is everything is fine. We can determine all the parameters pretty well. And therefore, we have good predictions of the model in order to test it. Yeah, there was a third possibility, mobilization, and this in the model scheme means that if this here is occupied, then it increases just this flux from the intracellular compartment. And the question now is, do we need this? And therefore, we did the typical model selection stuff. So here are the equations for our model I showed you in the first place, so the core model. These are the 6 populations, and it's just mass action kinetics with linear and bilinear terms. And if we now want to test whether we need this mobilization term, we just add one flux here that describes the mobilization of the Epo receptor if Epo is bound to the receptor. Our observables are these linear combinations or this component here with some scaling factor and some offset. So that means we have to do parameter estimation. We don't know all these parameters. And we have to do parameter estimation in this so-called partially observed dynamical system. And we have developed a lot of methods to do this in a reliable manner. Yeah, and the result is the following. We have one model with 10 parameters, the other has 11 parameters, so you can do the typical model selection either by likelihood ratio test, Aka-Ike, or Bayesian information criteria, and all 3 say the same, we do not need this. And for a dynamical system, it's also always nice to really look at the fluxes. So this is over time, the flux that is induced by the constitutive turnover. And the blue one here is the flux that is induced by this mobilization term. And if you take the area under the curve, for example, you see that this flux really does not play a certain rule. So that means we do not have to take this one into account. So that means these 2 mechanisms are at work. And before we figure out what are the effects of these 2 mechanisms, we want to validate, or we have to validate our model. And therefore, we do the following. So experimentally, it was only possible to observe these 2, in addition, this one and these 2. In the model, of course, each of the species has its own equation, and therefore it is trivial to really, by the model, look at the single populations here. And here on the left-hand side, you see the time dependence for the populations where the Epo receptor is involved, meaning this component, and this component. And here you have the Epo populations, meaning this one, and this one. So first looking at the Epo receptor population. So we have this component here, which was not measured at all. And what we see is that this, after stimulation, goes down pretty rapidly, but then recovers also pretty fast. So there is not really something like a period of being refractory. And this makes sense because this means that the system stays awake and is able to see new stimulations from the outside. And this is something that we can test experimentally. So sorry, this is one too much, but we can test experimentally. And this slide is perhaps a little bit busy, but I will go through it step by step. So this is a normal stimulation. You stimulate, Epo receptor gets activated, gets phosphorylated, and after a certain time it goes down. Now, if the system is not staying awake, that would mean if before we harvest the cells, if we re-stimulate with Epo, in the situation that it's not staying awake, the phosphorylation of the receptor would just go down as here. But if it stays awake, it would be possible to re-stimulate it and the phosphorylated Epo receptor concentration would go up. So, and here are the experimental data. In blue, the normal stimulation time course. Yeah, it goes up here around 20 minutes and then it goes down. And then if you re-stimulate, you see that at every point in time, we can re-stimulate the system, which validates our prediction of the system staying awake. So the second one is that we look at the EPO populations, and what was really surprising here is that the free EPO in the medium, that this is depleted relatively fast. And this also makes sense because if the system wants to see changes in the external concentration, it would be best to remove the current concentration of EPO as fast as possible. Yeah, so that means that the system can be pretty sensitive. And this prediction of the fast depletion of the external EPO can also be tested. So we did it in cell lines with the murine EPO receptor and the human EPO receptor, all these nice things biologists can do. And here you can now see the amount of EPO receptor, and it goes down in the same fashion as predicted. You can also look at this in vivo, slaughtering some mouse. Yeah, and here it goes down not in the same nice exponential-like manner, but it also declines pretty fast. So thereby we have validated by prediction of independent experiments our model so far, and now we want to figure out what's going on in the system. And therefore we took the model, and in the model simulation of the model, we observed two quantities. The first one is the amplitude of this Epo-EpoR receptor complex, and the other thing is the integral, the area under a curve, until a certain threshold is reached. And for the peak amplitude, we exactly see this kind of saturating behavior. But for the integral, we see a perfect linear relationship. And you can show mathematically that this is a linear relationship. Comparing it to experiments, here are the experimental data. For the amplitude, we see the saturation stuff. For the integral, we see the nice linear behavior. And if we go one step further down, so to the kinase that then triggers downstream signaling, we see the same event, same property here, the saturation for the amplitude and the linear relationship for the integral. So where comes this linearity from? And therefore, we again took the model and did an experiment in the model that cannot be done in the wet lab, meaning we played just around with this rate constant here. So if we take this recycling hypothesis, this was a value that was estimated from the data. And if we decrease it, or increase it, we see that the behavior still is linear, but only the slope changes. Yeah, that means linearity in itself does not depend on this process, but only the slope. On the other side, if we look at this constitutive turnover, here is the estimated value. And now if we decrease this, you see linearity breaks down. So therefore, it is this constitutive turnover that serves for the linear relationship, and the recycling just puts the slope on it. Yeah, this leads to a suggestion of another validation of the model. If we say that this process is important, then we can take a certain substance that blocks this transport. And then the question is what to measure to see how large the effect is. And therefore we did experimental design based on the model. So we simulated the whole story and looked which of the components are mostly affected by blocking this transport from the intracellular vesicles to the cell membrane. And it turned out that it is EPO that is most sensitive to changes here. And here is a simulation. So if we assume perfect blocking, then it just goes down a little bit and stays constant. That means that all EPO receptors that are present at the cell membrane are degraded, and it cannot process EPO, anymore. And then if we increase it, we get here, if we decrease the efficiency of the blocking, it gets down to this wild type situation. Now, if you do the experiment, you see exactly this effect. So you have here with blocking, and you can even estimate the efficiency of the blocking of the substance of this export, of this export here. Yeah, and based on these insights, we now want to go a step further and look at Epo and cancer. So as a drug, EPO not itself, so the EPO that our body is producing is applied, but a genetically modified EPO. The reason for this is that the body can produce it in a continuous fashion, but if you want to give it as a drug, you can only do it, or you don't want to do it in a continuous manner. And so therefore, this genetically modified EPO has a longer survival time in the blood system. And these modified EPOs are called E.S.A. for erythropoiesis stimulating agents. We will see this abbreviation again and again. And to come to cancer, chemotherapy damages cells with a high turnover. This is a basic idea. And since our system has also a high turnover, it also damaged the blood building system. Yeah, and then people get an anemia. And so therefore these E.S.As are often applied during chemotherapy to fight anemia. But now the first problem arises. Often EPO receptors are also expressed on tumor cells. And of course, tumor cells do not turn into red blood cells when they see EPO, but I will show you on the next slide what these guys do when they see EPO. And on the other side, there's a risk of overdosing that lead to cardiovascular events. If you get too much EPO, you produce too much red blood cells, and so to speak, your blood gets thick, and this can then lead to these thrombovascular effects. Yeah? So the idea now is that we want to determine a minimal but effective dosing scheme in order not to trigger the effect I show you on the next slide and not to trigger these adverse effects. So EPO counteracts chemotherapy. In these 3 slides, what is shown over time is the viability of cells, meaning how happy they are. And if we take, for example, here one cancer cell line, and we look at the growth of these cell lines without EPO and with EPO, you see there's no difference. Then if we apply cisplatin, which is the therapeuticum, chemotherapeuticum, you see without EPO, the cells are dying, and EPO can rescue this effect. If we now take a certain variant of this cell with overexpressed human EPO receptor, you see in this situation without treatment, they behave the same. And here EPO leads to increase of these cells. And you can also pinpoint this down on a more molecular mechanism. Jim yesterday spoke about caspases. And so if you look at caspase activity here and only look at these colors here, so if we just apply cisplatin, then apoptosis gets activated, and the more EPO we put on the cells, the lesser these curves are, and this is what rescues the cells by the end of the day. So now coming to the treatment of the patients. So there is a substance that's called C.E.R.A. It's a drug from Roche. C.E.R.A. stands for continuous erythropoietin receptor activation, and this is one of these E.S.As. And we look in the following at a certain time of cancer. And the story behind this is that Roche has approved this drug for the treatment of anemia in case of chronic kidney disease. As I mentioned on the very first slide, in the kidney, the EPO is produced, and therefore if you have severe problems with your kidney, there were no blood cells to produce, and all these people get these artificial EPO substitute diseases. And so Roche a few years ago said, okay, let's get improvement for this drug also for chemotherapy-associated anemia. And they did 2 large clinical trials, and both were terminated, not at the end, but preliminary, because both failed for 2 reasons: increase of the tumors and cardiovascular effects, exactly the 2 phenomena I mentioned before. So in the following, we will measure the amount of red blood cells by hemoglobin. So this is proportional to the number of red blood cells. And if you have an HB value of 12, you are fine. If you are below 10, you get an anemia. If you are above 15, you get risk of cardiovascular events. So now we extended our model on the cell level by a pharmacokinetic and pharmacodynamic model. So pharmacokinetics means what is the body doing to the drug, and this means essentially how from the subcutaneous injection here it goes to the drug, sorry, it goes to the blood and then goes to the cell. And then there's a pharmacodynamic part that means what the drug is doing to the body, and this here means essentially that HB, that red blood cells are produced. So here are two data from these clinical studies. This is a healthy subject, healthy control, and this is a patient. Every 2 weeks, there was an injection with this C.E.R.A substance, and then C.E.R.A levels in the blood were measured, and HB levels were measured. Yeah, and you can see here, this healthy subject, the HB level also was a little bit increased, and giving C.E.R.A, it even increases more to a somehow dangerous situation. Here the patient, every 3 weeks, here the C.E.R.A levels, and you can see he was in the beginning or she in an anemic situation, and this is more or less cured during the time. And the straight lines here are fits, and the shaded areas are error bars from the fits. So it turned out that the biochemistry in itself, so the model of on the cellular level, that this is not dependent on the patient. This makes sense. And by a lot of statistical testing, we figured out that in our model, we only need 2 patient-specific parameters. That is a number of the ESA binding site, and this corresponds to the number of these progenitor cells, and the hemoglobin degradation rate. And this also makes sense from a biological point of view, because cancer usually comes with inflammatory effects, with inflammation, and inflammation decreases the lifetime of the red blood cells. So if we do this for around 100 patients and 80 controls, we here have the distribution of the number of red blood cells, these ESA binding sites, and the degradation rate. And you can see green is healthy, purple are the patients, that the number of binding sites, the logarithmic scale here, the number of binding sites in the mean is smaller, and especially, and this was somehow surprising, that the degradation rate is increased by an order of magnitude. So if you want to treat the patients, you have 2 opposing goals. You want to increase the HB level as soon as possible to treat the patients, to treat anemia. But if you do it too rapidly, the people are not feeling well. And I will show you what this means in a graph in a second. And of course, you want to avoid the overdosing. And right now, the scheme that is applied in the hospital is very simple. So you start treating with a certain dose. If you are below 6 grams per deciliter, then hopefully HB level increases. You halve the dose if you reach 12, and you stop treating if you're at 13. Then typically HB levels go down, and then you restart with 25%, and then if it goes even further down, you continue. So this is a simple scheme. So if you now apply this scheme, and these are real patients, if you apply this scheme, here is a patient that has a very small degradation rate of HB. It gets 2 doses after the scheme, and you see he is heavily overdosed. Yeah, so this is a candidate for cardiovascular problems. And here I also show you this kind of not too fast increase. Yeah, so there is a certain slope that should be followed to get to the value of 12. If you do it this way, people feel very unhappy. The second one, with a medium degradation rate, and you see here it works out more or less perfectly. Yeah, and here's a patient with a very high degradation rate, and you see he is not responding at all and need blood transfusion here in order to survive. So this is a complete cohort of patients that we had. This was the first patient that I show you. This was the responder, and this was a non-responder. And what was also kind of surprising is that only based on the degradation rate, yeah, you can pretty well discriminate these 3 groups of patients. But you also see that there are a lot of people who are overdosed and a lot of people who are not responding. And so now we took the model and somehow inverted it to find the optimal treatment strategy in order to reach the desired temporal evolution of the HB value. Yeah, and for this guy where we had the large overshoot in the beginning, it now behaves perfectly. This one was already pretty good, it stays pretty good. And here for the non-responder, we could also follow this curve, but of course the the amount of drug that has to be given has to be much larger than in the 2 other cases. And if we do this for all the patients, then we can treat everyone with a certain range of doses, and typically it's a 3-week treatment, and for these very severe patients, we then need a weekly treatment, but we get all of them responding. This was, of course, an offline procedure, but we can also turn this into an online procedure. And therefore, we collect data, and with each data point, we re-estimate the parameters of the system. And since we get more and more data points, prediction of the model will become better and better. So if we start with one data point, with some initial guesses for the parameters, we see here a huge uncertainty, meaning we more or less don't know anything. And then we update these parameters, and you will see that this area gets smaller and smaller. And as soon as we cross here the level of 10, which is the critical one, then we again, based on the model, do a treatment in order to get this kind, you will see it in a second, behavior of rescuing the patient. These are now simulated data. I will show you real world data afterwards. And the problem, or one problem, is that the measurements here, they have an error of 1. Yeah, so they are pretty noisy. And so therefore, if the true value is here, but the noise realization brings it there, the medical doctor would say, treat it now, but the model can, of course, look beneath this noise and can do a more reliable prediction. So, and now we let time run. Yeah, things develop. You see, the more data we get. Yeah, here we have exactly the situation I mentioned before. This is roughly below 10. Model says no problem so far, and we are still from the model in this pre-anemic situation, and then things get worse. And after a certain point in time, now the model is below 10 and says we have to do something, and that means we have now a plan for a treatment. So we get a standard dose at the beginning, and based on the standard dose, we hope to follow this trajectory, but the model says it will go this direction. And therefore, we can plan a treatment to get the black model curve closer to the green desired curve. Yeah, and this is how it looks like here. So then we let the model run and learn, and you see the suggestions for treatment, they change with every new data point. And from here to, so here we had large treatment, but the patient behaves better than expected. And we always rearrange the parameters, refit the parameters, and then you can let it run. And after a certain point in time, after a certain point in time, anemia is cured. So, and this can also be, and this is the plan in the long run, really transferred to a tablet for the physicians. So, and these are real world data, though this is a patient. And this today was, when we built this model, this was really the day we did it. And now in this model, for this software, for this application, you can now do planning of the treatment. So you can give anemia, you can give blood transfusion, and you can take into account that you do chemotherapy. So for example, the model says if I do chemotherapy here in this time window, so this is time, yeah, then it will go down like this, and this would be a region where it's critical. And so you can think about what is happening if we apply anemia or if we do blood transfusion, and you can of course also look at the combination end of this and thereby try to design an optimal treatment for the patient. Yeah, and with this, I would like to thank all the people. So it is a long-term collaboration with the group of Ursula Klingmüller. It's a German Cancer Research Center. And we had, of course, hospitals involved here, and are also very happy that we have this collaboration with Roche. Otherwise, we would not have gotten these interesting data. Yeah, and people from my group, of course. They supported the study. So to summarize, in the first part, we saw how the Epo receptor is processing this information of spanning potentially 3 orders of magnitude. We saw that the fast recovery of the cell surface Epo receptors helps the system to stay awake, that the rapid depletion of Epo makes it possible to determine the outside concentration in a reliable way. And we saw that we have this linear relationship between the outside Epo levels and the integral of the Epo receptor activation, and this allows for pretty accurate translation of the ligand input, and then in combining it with a pharmacokinetic and pharmacodynamic model, we could see that we can stratify patients by just looking at one of these patient individual parameters, degradation rate of the red blood cells, of the hemoglobin, and we can have seen that we can use this for an optimal treatment of anemia in single patients. And with this, I thank you for your attention. And I'm happy to take any questions.
Thank you, Jens. So we have time for maybe short questions, not to wait too much. Peter? I think your micro is off.
Thanks. I just had a question on this linear relationship that you also have here on the summary slide, right? So you mentioned that this turnover is critical for sustaining basically this linear relationship. I think I can imagine how this works, but I don't think you explained it. Could you briefly summarize this idea?
Yeah, the point is if the turnover is not fast enough, yeah, then you cannot see the larger doses.
Yeah.
Yeah, because you don't have enough EPO receptors there.
Yeah. No, because it's the degradation, right? That was important. No, there's these extra variables that you, or these extra pathways that are necessary for this linear relationship between this integral and.
Sorry. This was easier when we had overheads. So
yeah, this one, right? So the circled pathways. These are.
Yeah. So if you increase the extracellular concentration, then you need more EPO receptors on the cell surface, and therefore the turnover. Yeah, here we decrease this rate constant. Yeah, and so therefore the system just saturates.
No, exactly.
Yeah,
very clear. Yeah.
Okay. Thanks for the question.
Okay, Geneviève, also the question.
Okay, thank you for this very nice talk, very interesting. So I have a quite general question. So I would like to know how, I guess other receptors have a similar behavior. So I would like to know if you could comment on the possible generality of this type of model.
What was the first part of your question, of your sentence? So
just, I guess that other, so when I see the scheme, the general scheme with the internalization and so on, it's quite common.
Yeah.
I think, so I just wonder if other receptors could be described by similar models.
Yeah. So the scheme is pretty general. But for example, if you look at the EGF receptor, they are expressed in the order of 100,000 receptors per cell. Yeah, so it's completely different order of magnitude. And so therefore, this system could see huge differences without the trick the EPO receptor has developed. Yeah, but in general, these internalization and externalization, this is, and the degradation of the ligand, this is pretty general. Yeah, this applies also, for example, to the EGF receptor, but the amounts of receptor are completely different, and therefore the way the information process is going on might be different. But the scheme is pretty general.
Okay.